Hello dear students, today we are going to discuss the topic Cryptochromes, their discovery, chemical nature, role in photomorphogenesis and mode of action. The main objectives of today's topic are: number 1, to get aware about the blue light photoreceptor cryptochrome. Number 2, to study the discovery of cryptochromes in detail. Number 3, to get familiar with the chemical nature of cryptochromes. Number 4, to understand the mechanism of action of cryptochromes. And finally, to have an idea of the role that cryptochromes play in photomorphogenesis. First of all, let's see what cryptochromes are. Cryptochromes are flavoprotein blue light photoreceptors. They are found in many organisms including bacteria, plants, animals and humans. They mediate a variety of blue light dependent responses including photomorphogenesis and growth responses in plants. They also function in entrainment of the circadian clock in plants and animals such as Drosophila and mouse. Unlike phytochrome induced responses in plants, cryptochrome induced responses are not photoreversible. The cryptochrome induced responses, however, become apparent more rapidly when compared to phytochrome based responses. Cryptochromes are the evolutionary descendants of DNA photolyases. DNA photolyases are DNA repair enzymes which act in a light dependent manner. These blue and ultraviolet light receptors share sequence similarity to DNA photolyases. DNA photolyases help in removing UV-induced pyrimidine dimers in DNA of microbes by utilizing blue light. But in spite of sequence similarity to DNA photolyases, cryptochromes have no photolyase activity. Cryptochromes are widely distributed in bacteria and eukaryotes, but absent in archaea. Quite recently, they have been found in various animal lineages, including insects, fish, amphibians, and mammals. Now let's move ahead with a brief discovery of cryptochromes. Up to 1990s, nothing was known about the blue light receptors of plants. However, in the past 5 decades, some progress was reported about the identification of mutants for blue light responses. These were subsequently followed by isolation and characterization of CRY mutants. For example, the HY4 or CRY1 mutant of Arabidopsis was isolated and characterized. It lacks the blue light stimulated inhibition of hypocotyl elongation. At the same time, it was found to encode a 75 kDa protein having significant homology with the microbial DNA photolyase. In view of its similarity to the microbial DNA photolyase, the HY4 protein was considered to be the blue light receptor inhibiting stem elongation. This blue light photoreceptor was later renamed as CRY1 or Cryptochrome 1 by Grizzle in 1979. Subsequently, in the year 1996, another blue light receptor, CRY2, was identified in Arabidopsis by Lin and co-workers. The CRY2 protein was also found to induce light-dependent hypocotyl growth inhibition. Cryptochromes have been found in dicots like Arabidopsis, tomato, and in some monocots like rice, barley, and others. Most plant species studied contain multiple cryptochromes. For example, Arabidopsis has 2 cryptochrome genes, CRY1 and CRY2. Tomato and barley each have at least 3 cryptochrome genes, CRY1A, CRY1B, and CRY2. In addition to higher plants, cryptochromes were discovered recently in some alga, ferns, and mosses. In the fern Adiantum sapulus venereus, 5 cryptochrome genes were found to influence its spore germination in response to blue light. In the moss Physcometrella patterns, two cryptochrome genes were found to control oxygen sensitivity in a blue light specific manner. In the green alga Chlamydomonas, a rhodopsin-like photoreceptor residing in the eye spot was discovered and speculated to control blue light responses in this algae. Now, the discovery of cryptochromes in animals. In the first approach, mutant was identified in a screen involving transgenic flies. These transgenic flies were altered to express a luciferase reporter gene, which was linked to the promoter of period gene. The period gene performs an integral role in the central oscillator of the Drosophila circadian clock. This CRY mutant exhibited arrhythmic luciferase expression. In the second approach, the gene was identified as encoding a protein having sequence similarity to both photolyases and the mammalian and Arabidopsis CRY sequences. This led to the idea that this gene encodes a Drosophila blue light photoreceptor. Now, a brief account of discovery of cryptochromes in mammals. Mammalian cryptochromes was identified by screening of human cDNA library. Cryptochromes were found to have sequence similarities with the Drosophila photolyase genes. As of now, cryptochromes have been identified in a variety of animals including zebrafish. Now, something about the discovery of bacterial and related cryptochromes. Cryptochromes were previously thought to be restricted to eukaryotic organisms only. However, they have also been identified from some bacteria. In Synechocystis, photolyase-like sequence was identified which lacks photolyase activity. This photolyase-like protein was later designated as a cryptochrome. This cryptochrome sequence was named as CRY-. Now, the second important aspect of cryptochromes is their chemical nature. The biochemical nature of blue light absorbing chromophores has been elucidated quite recently. A.W. Galston suggested nearly half a century ago that flavin was the most likely chromophore. However, zeaxanthin has also been speculated to be responsible for some of the blue light induced responses. The flavin carotenoid controversy was difficult to resolve because cryptochrome mediated responses are not photoreversible. Because of its hidden nature, the pigment was called a cryptochrome by Grizzle in 1979. Cryptochrome appears to be similar to DNA photolyase in terms of its overall structure. Their similarity indicates an evolutionary relationship between the two. As such, it has been suggested that microbial photolyases are the precursors of cryptochromes. However, the two differ in some features. Firstly, cryptochromes have a distinguishing carboxy terminal extension, which is not found in photolyases. The second difference is that cryptochromes show no photolyase activity. Cryptochromes are composed of two domains, an amino-terminal photolyase-related region that shares sequence homology with photolyases and a carboxy-terminal domain that has no strong sequence similarity to known protein domains. PHR contains two chromophores, a light harvesting chromophore which is either a folate, methynyl tetrahydrofolate or a diazoflavin, and a catalytic chromophore that is flavin adenine dinucleotide FAD. The carboxy terminal side of the protein, on the other hand, is less conserved than the PHR region. It is relatively longer in most of the plant cryptochromes than the animal cryptochromes. The PHR region have remarkably similar three-dimensional structures. They are comprised of two domains, including an alpha-beta domain and a helical domain. These domains are connected by a loop-like structure that wraps around the alpha-beta domain. Almost all residues known to be important for chromophore binding in photolyase are conserved in cryptochromes. On the other hand, residues of photolyase critical for binding of DNA ligands and role in repair are not equally conserved in cryptochromes. It was found in E. coli that the CRY1 protein, in addition to binding FAD, also binds to pterin. Pterins are light-absorbing pterin derivatives that often function as pigments in insects, fishes and birds. This suggests the possible role of pterins as one of the chromophores of blue light receptor cryptochrome. Let's move now towards the mechanism of action of cryptochromes. In the last few years, significant progress has been made in understanding the mode of action of cryptochromes. The blue light excited cryptochrome molecules undergo a number of biophysical and biochemical changes. These changes mainly include electron transfer, phosphorylation and ubiquitination. This ultimately results in conformational change of cryptochrome molecules and hence transduction of the light signals to the downstream transcription factors. Two modes of cryptochrome action are known till now. One of these include cryptochrome interacting basic-helix-loop-helix 1 dependent CRY2 regulation of transcription. The second one is suppressor of PHA constitutively photomorphogenic dependent cryptochrome regulation of proteolysis. These mechanisms depend on blue light dependent interactions between cryptochrome and its downstream signaling proteins. This leads to modulation of gene expression and ultimately altered developmental programs in plants. Cryptochromes mainly accumulate in the nucleus, but some of the cryptochrome mediated action in response to blue light also occur in the cytoplasm. This was confirmed by the isolation of CRY1 mutant seedlings, which showed impaired activation of anion channels at the plasma membrane. Of the two domains of cryptochrome, the carboxy-terminal domain plays a role in the function and regulation of both animal and plant cryptochromes. This has been demonstrated in cryptochrome of Arabidopsis, which showed constitutive expression of carboxy-terminal domains even in the absence of light. This led to a constitutive growth response to light even in darkness and in absence of the PHR region. However, this is not always the case, as the PHR regions of the Drosophila and Xenopus cryptochromes are physiologically active when the carboxy-terminal domain is absent. The carboxy-terminal domain of Drosophila cry is, however, important for stability of the protein. It has also been linked to its sensitivity to the circadian light signals. In case of Xenopus, the carboxy-terminal domain of the cry is required for its nuclear localization. Now let's see how cryptochromes are regulated. Phosphorylation is one of the mechanisms involved in the regulation of cryptochromes. The Arabidopsis cryptochromes are phosphorylated in response to blue light. This is associated with the function and regulation of the photoreceptor. Moreover, CRY1 cryptochrome in insect cells was found to undergo ATP and blue light dependent autophosphorylation. However, till date, it is not known whether animal cryptochromes also bind to ATP. The exception lies with the mouse cryptochromes, which are phosphorylated. Now finally, role of cryptochromes in photomorphogenesis. The role of light in regulating morphogenesis is known as photomorphogenesis. Red and blue light both are essential for morphogenetic development in plants. Some of the most important responses in plants which occur in response to blue light are as: Number 1. Role of cryptochrome in blue light inhibition of hypocotyl elongation. A dicot plant, when germinated in dark, develops an etiolated seedling with a rapidly elongated hypocotyls. This allows the unopened cotyledons containing no photosynthetically competent chloroplasts to emerge rapidly from soil. Exposure to light results in de-etiolation or photomorphogenesis. The de-etiolation response include inhibition of hypocotyl elongation, stimulation of cotyledon opening, change of gene expression, and induction of chloroplast development. Because the hypocotyl inhibition response is easy to monitor and measure, it has become the most widely used assay in the study of cryptochromes. Arabidopsis CRY1 was identified on the basis of the study of the HY4 mutant impaired in blue light inhibition of hypocotyl elongation. When grown in blue light, HY4 mutants showed a significantly reduced blue light inhibition of hypocotyl elongation, resulting from mutations of the CRY1 gene. Transgenic tobacco or Arabidopsis seedlings overexpressing Arabidopsis CRY1 had hypocotyls shorter than those of the wild type when grown in blue light. It indicates that the amount of CRY1 is rate limiting in the process of blue light inhibition of hypocotyl elongation. Transgenic Arabidopsis plants overexpressing CRY2 also showed a short hypocotyl phenotype in blue light, which led to the hypothesis that CRY2 is also involved in hypocotyl inhibition. Furthermore, it was found that the CRY1-CRY2 double mutant exhibited a more pronounced long hypocotyl phenotype when grown in continuous blue light than the CRY1 or CRY2 monogenic mutant. This suggests a partially redundant function of the two cryptochromes in this response. Number 2. Role of cryptochrome in blue light stimulation of cotyledon expansion. In contrast to the inhibitory effect of cryptochromes on cell elongation in hypocotyls, cryptochromes mediate blue light stimulation of cell expansion in cotyledons. Both CRY1 and CRY2 mediate blue light stimulation of cotyledon expansion. As in the case of hypocotyl inhibition, CRY1 stimulates cotyledon expansion in both high and low irradiance, whereas the role of CRY2 is limited to low irradiance. Transgenic seedlings overexpressing either CRY1 or CRY2 show larger cotyledons than those of wild-type seedlings grown under blue light, suggesting that the cellular concentration of cryptochromes is rate limiting for cotyledon expansion. The contrasting effect of cryptochromes in inhibition of hypocotyl and cotyledon expansion suggests that different signaling mechanisms are involved in two organs. Number 3. Role of cryptochrome in chloroplast development. The major mechanism behind chloroplast development during deethylation is the change in gene expression in both nuclear and plastid genomes. Cryptochromes mediate blue light induction of nuclear genes. These genes encode proteins which are essential for photosynthesis and other functions of plastids. Some of these genes include CAB, RBCS, and CHS, which also form components of the plastid transcriptional apparatus. CRY1 is the major photoreceptor that mediates this induction of gene expression of nuclear genes, particularly SIG5. The cryptochrome helps in supplying the nuclear encoded proteins to the plastid. These proteins play an important role in the transcription that occurs in chloroplast during deethylation. Number 4. Function of cryptochrome in the control of flowering time. Plant flowering time is controlled by various signal transduction cascades. These cascades connect many environmental signals to developmental programs. One of the most important environmental signals affecting flowering time is day length or the photoperiod. Although it is well known that phytochromes are major photoreceptors regulating flowering time, but a role for cryptochrome in photoperiodic response has been shown recently. The function of cryptochrome in the flowering time has been studied in Arabidopsis photoreceptor mutants. The role of Arabidopsis CRY1 in promoting floral initiation has been demonstrated by studies showing CRY1 mutants flowered later than the wild type in various light conditions. The function of Arabidopsis CRY2 in flowering time control has also been studied using the CRY2 mutant. The CRY2 mutant flowers later than the wild type in long day but not in short day, whereas transgenic plants overexpressing CRY2 flowered slightly early in short day but not in a long day. Therefore, either a mutation or an overexpression of the CRY2 gene resulted in the reduced sensitivity to photoperiods. Some additional mechanisms or pathways are also employed by cryptochromes to affect the flowering time of plant. For example, the clock-dependent transcription of constans gene and other flowering time genes are altered by the activity of cryptochrome 1 and cryptochrome 2. Both CRY1 and CRY2 have also been shown to mediate the expression of microRNA 172. This microRNA has been found to modulate flowering time independently of the constans protein. Number 5. Role of cryptochrome in entrainment of the circadian clock. The circadian clock is the molecular oscillator that drives various apparent circadian rhythms in most organisms. This allows the organisms to anticipate and adapt to environmental changes both on daily basis as well as the annual photoperiod cycles on Earth. In 1998, cryptochrome was found to act as a major photoreceptor of the circadian clock in Arabidopsis, Drosophila and mouse. In Arabidopsis, this was confirmed from the impaired circadian clock of the CRY mutants. As for example, the CRY1 mutant showed lengthened period of the circadian rhythms in both high and low fluence rates of blue light. On the other hand, the CRY mutant has a slightly shortened period in low fluence rates of blue light. The CRY1 CRY2 double mutant showed a longer period under the fluence rates of blue light. This suggests that Arabidopsis CRY1 and CRY2 act redundantly to entrain the circadian clock. However, in case of mouse CRY1 CRY2 double mutant, the circadian clock becomes arrhythmic. In short, it seems that cryptochromes are not intrinsic components of the circadian oscillator in Arabidopsis. Number 6. Role of cryptochrome in stomatal opening. Phototropin were considered as the sole photoreceptors which regulate stomatal opening. A recent observation showed that cryptochromes also contribute to the blue light stimulation of stomatal opening. It was found that the CRY1 CRY2 mutant and CRY1 gene overexpressing plants exhibit a reduced or increased stomata opening in response to blue light respectively. This was all about today's topic. Thank you.